So again, welcome to this webinar on deconvolution with Lightsheet. My name is Nicolaas van der Voort. I'm Director of Scientific Affairs, and it's a pleasure to see so many familiar names popping up in the chat. Today, we're going to cover deconvolution and of Lightsheet images. And a Lightsheet is a pretty varied field because there's many different systems, many different types of doing Lightsheet. So we'll try to sort of paint a spectrum of the different options and, of course, how to handle that in Huygens. And this meeting is being recorded so that we can make it available to you afterward. If you have any question during the webinar, you can pop it in the chat and we'll try to keep a check of that and answer. And afterwards, we'll have a Q&A session that won't be recorded. So feel free to unmute yourself over there. So we'll be covering today Gaussian type light sheets, and these are typically used for, these have a spatially varying PSF, and they're used for big data. We will also be covering flat types, that's either lattice light sheet or for another type of flat light sheet. And these are of a different type, so we will cover both, and we will also deal with fusion. Now we'll do 20 minutes presentation and then 15 minutes demonstration roughly. So the demonstration will show a new visual aid of setting the light sheet parameters for the deconvolution. We will be covering the deconvolution itself, and we'll be showing what's new for handling big data. We will not be covering associated topics, which include stitching, de-skewing, visualization, and analysis. So these you can check other webinars for that I will link in the description. So this is focused on light sheet deconvolution. So to kick things off, let's consider a regular Gaussian type of light sheet. So in this case, I am focusing a Gaussian beam, and in the center, it is narrow, and that gives me a good Z resolution. But away from the center, the beam gets wider, and that causes the Z resolution to deteriorate. Now, you also have scattering to make things more complex. So this gives you an effective range, which is related to the Rayleigh range over which your resolution is good. So what deconvolution does for the Gaussian type is it is very good at increasing the Z resolution over the whole Rayleigh range. Whereas before you may only have been able to use a small part, you can very effectively increase the Z resolution, whereby you have a much broader view and have a much broader range over which you have a good resolution. So I brought some data to show this. This is data from Alex Wong from HMS. And this is the raw data, and this axis is the Z-axis, and this is the X-axis. So the vertical axis is the Z-axis. And what you can see here in the middle is exactly this. In the middle, the Z resolution is decent. But if you move away, then you can see all these sort of elongated shapes appearing, which are indicative of the of this effect with the Gaussian extending the Z resolution, of extending the Z PSF. So the deconvolution is very good at counteracting, like you can see here on the bottom. And this XY, so this is now XY image, shows that in more detail. So we have these structures, and these are some kind of channels. And within these channels, we have smaller channels, and you can beautifully see that. And what you also see here is that in the raw data, because all the Z PSFs are elongated, it becomes very crowded. But after the deconvolution, when the Z resolution is much better, then now we see only the structures that correspond to that particular plane. And this was done on a movie SPIM commercial type of microscope. And so if you do all of that, of course, we set the right deconvolution, we set the right metadata, deconvolution, Then these were 10 tiles stitched, vignetting corrected, and this is then a surface rendering overlaid with a maximum intensity projection of the whole image. So the Gaussian family of light sheet comes in, we have 4 types of them in Huygens, of which the first 2 are most important. So we have the regular Gaussian light sheet, which assumes 1 illumination objective. And there is then also the multi-view light sheet, which is similar, except it has 2 illumination objectives, and there's some alterations for the deconvolution to take that into account. And most commercial systems nowadays have multiple illuminations. So you'll find a lot of different commercial systems in this category, and we've experienced with very many of them. Also, you'll often find this in a configuration where the objectives are oblique with respect to the cover slip, in which case you additionally need de-skewing as well. Now, here on the right, we have 2 variants that are less common. Essentially, the high fill factor is when you overfill the illumination objective, and that makes your light sheet more like an airy disk and less like a Gaussian. And we also have a model for the cylindrical lens. So these 2 are less common, so we'll forget about them going further. So it's really these 2 on the left that you will encounter quite commonly. And of course, Huygens then has the spatially varying PSF. So Huygens makes the view into many smaller views, has a different PSF for each, and thereby it can correct the effect of the Gaussian, the variable PSF along the Gaussian. Now, in addition to deconvolution, there's quite a number of cases where you want to use fusion. And I will enumerate these. So in the case where we have single illumination and single detection, you do deconvolution. Most of the time you also do stitching, but you don't need fusion. Right. Now, if I add another illumination objective, like with the movie or the multiple illumination, then you can choose to have alternating single illumination or simultaneous double illumination. So if you go for alternating single illumination, you want to do the deconvolution, potentially stitching, followed by fusion. Now, the alternating single illumination has an advantage that you don't need to align your illumination beams. This can be really an experiment. It can be tricky to manage this experimentally. And so instead of aligning the 2 illumination beams precisely, you can just record 2 single views and the alignment will be done in the Huygens fuser. So if you do the simultaneous illumination, then you don't need fusion because you only have 1 view. Then there is the case where there's 2 detection objectives, and this is notably the Leica system. You want to use fusion to fuse the views of the 2 objectives. And lastly, if you're rotating your sample, and this is historically how the earlier light sheet systems did it, the sample was rotated, and in this way, multiple views are recorded, and then you want to use fusion as well. So here's an example. This is a zebrafish by Denis Resnikov. And sorry, it's not, it's a Drosophila brain. And so you can see 8 different views being recorded at 45-degree angles. And these are then beautifully fused and deconvolved in Huygens. And if you'd like to know more about that, then this is the link to the full webinar describing that. This is an example we see more of in recent times. So we have a cellular organoid from HeLa cells. On the left is with fusion, on the right is without, and you characteristically see that on the right part of the image is missing because it hasn't been illuminated. And this is with a Lavision ultrascope from Stelias Didascalou, by the way. Okay, so moving on to the next category of light sheets. We have the flat types. And these again fall into two categories. So we have the flat Gaussian types, and we have the lattice types of light sheets. So for the flat Gaussian, there's a lot of development going on into hardware solutions of making the Gaussian light sheet flat. And these usually involve scanning the focus of the illumination sheet and timing that in various configurations with the camera, such that each time you have the narrow side of the light sheet, the narrow part. So it's typically called actually swept light sheet with a rolling shutter. There's the Mesospim initiative that uses this principle, and many manufacturers as well. So this is also, again, large category. So here on the right, we have the lattice light sheet types. So we have the bezel beam, and so that has the annular ring for the illumination profile, or the scanning bezel lattice with the engineered PSF. And Huygens has all of these PSFs by default, and is also changing the PSF along the propagation direction. Although, of course, for these types, the engineered to change much less than the Gaussian type. So the effect is less noticeable. If you have a custom pattern, you can use an experimental PSF. So if you have another custom engineered PSF for your light sheet, you can use the measured PSF. And this configuration also commonly occurs in the oblique state. Now, I'd like to share a customer story for the lattice light sheet, and this was with Stephen Edwards and Hans Blöm from University of Stockholm. And what they did is they wanted to see how fast and for how long times you can look at live processes in cells. In this case, it's ER and mitochondria dynamics. And this is an impression of a single time frame for this image. And so for the lattice light sheet specifically, it sort of occupies the space in the microscopy field where it has a high temporal resolution, high sample health, and but for this recording, spatial resolution is decent, but the SNR suffers. And so with the deconvolution, we're going to see how we can expand this pyramid of compromise. We're going to get higher spatial resolution, and we're going to get higher SNR. And this is actually part of a whole workflow that we also discussed in more detail, for example, during the BINA Lattice Light Sheet user group. De-skewing, deconvolution, stabilization, rotation, visualization, tracking, and colocalization. Today, we focus only on the deconvolution, but it's part of a whole workflow. So this is the mitochondrial channel result, and with compression applied. And what you can see here is that also it's well sampled. These structures are then nicely resolved, this mitochondrial structure. You can get a good resolution out. And the ER channel is actually more challenging and perhaps therefore more interesting because it consists of alternating sheets and tubes or lines, if you will. And so there's a lot of heterogeneity in this structure. And for example, if you look here within the white circle, this is more like a sheet. And indeed, Huygens conserves a sheet. Whereas here, if you look in the yellow circle, then we see that these are more tubes with hollow spaces around them. And Huygens is very well able to obtain these results. And the same goes here for the bottom. These are more examples of the spatial accuracy. And this is then the result overlay, so you can nicely see that the ER is around the mitochondria. And this is then the picture of that image moving with a very high time resolution, right? So if you want to capture very fast processes, but you want to capture them for a long time, you fight against photo bleaching. This is a very beautiful result of pushing the limits on those frontiers. And this just slightly off topic, but this is with a tracking analysis. And here, if you follow this guy here, this mitochondria is presumably being transported. It's moving along the direction of the white arrows, and you can do tracking analysis as well. So to clarify a little bit what options are associated with what license options you need to do this analysis. So there is the light sheet deconvolution option, and that covers all of the light sheet types that we discussed today. So additionally, you typically want to have the stitching option. We didn't discuss it, but if you want to use stitching, you need the stitching option. And then there is the fusion option if you want to do fusion. Now, additionally, you want to have adequate performance. Adequate depends a little bit. We're happy to advise you, but if you're handling big data, it helps if you have a big computer and a license to back that up. We're adding pyramid files, which I will show in my demo, and that will be added to the Huygens Professional. So Lightsheet is a very professional topic. If you're on the Essential, you may consider upgrading to the Professional to get access to the pyramids. And if you need de-skewing, you need the stabilizer option. Now there's more depending on your application, but these are really the important ones. So we're happy to check with you which ones of these you have and which you haven't yet in case there's any confusion. Okay, so I already mentioned we have the fusion webinar, we have the stitching webinar online, and 21st of October, by then we'll have our new version released. We'll do the What's New webinar covering pyramids in more detail, cross hot pixel correction, and the new Mac version, and much more. So stay tuned for that. So let's go to the demonstration. If you have any questions in the meantime, don't hesitate to ask. I will keep an eye on the chat and answer them. So this is the beta version of the 2510 for Huygens. And this image here, let me just make it bigger, like so. This image is corresponding to the first data set that I showed from human colon. And now the first thing I'm going to do is I'm going to make a crop. And that I do because I will use different parameters. I will maybe try different settings to see which one works best. And then it's just so much easier if my image is small, because then I don't have to wait so long. So I'm going to make what I call a pencil crop. So you'll notice that I'm not cropping it along the X direction, because the X direction is the direction of the light sheet. So I'm cropping it in Z, and I'm cropping it in Y. And I'm also, for this demonstration, only interested in channels 0 and 2. So I'm disabling the other ones. I'm going to click Extract, which creates a copy of the image that I've just selected. So it's this one. So this is the crop that I just made. So I'm now going to show you, so in order to do the light sheet deconvolution, you need to get a few technical parameters right. And we want to make this more transparent and easier to use, so we develop a new tool for that. So in this window, you have here the general pixel sizes, the emission, excitation wavelengths. These things are clear. But now here for the light sheet specific parameters. By the way, these are spherical aberration associated. We don't need them right now. So I'm going to launch the new light sheet parameter tool. And this is still under development, so it's probably not going to be part of the initial 2510 release, but we'll release it soon afterwards. So it's still work in progress. So I have my two channels, and here at the top, I see these different types that I was just discussing in my presentation. So if I choose Gaussian, I see one illumination, one detection objective, and I can choose if it's from the left or from the right. If I choose multi-view and I choose right and left, then I get my two illumination objectives. So you can already visually confirm that the configuration of the objectives are in fact what you know them to be from your setup. Now, if we go on, then we have to set the width of the light sheet. And there's always two definitions. There's the full width F max definition, and there's the 1/E squared definition. The width is typically referring to the 1/E squared definition. So to avoid any confusion, we report them here both. They're related by a factor 1.7. And if I update this number, then automatically the other number changes as well. So if you know either of them, you can fill that in, and that works. And also this illustration, so this beam profile is now also changing. So I know this value to be 1.9, so I add that, and I can also visually check that that corresponds with the PSFs that I am seeing. Now, Huygens by default assumes that the center of the light sheet is in the center of the image. But for this image, it's not the case. So we can see actually that the brightest and best resolution is here. So it's focused more towards the center of the object. So so I just drag it, and you see this number updates to -300, and that's set. So that's great. There's a question: do we need to supply .mvl file with the Zeiss LS7 data for multi-angle fusion metadata? I apologize, I have to pass that one on to my colleagues. We have different ways of reading in the positional metadata from the different views. And I will forward that question to my colleagues, and you'll have your answer next working day here in the Netherlands. I'm just not 100% sure which metadata we read in for that, but we have multiple ways for doing that. So what you'll notice as well is there's this dot here, and this dot indicates the focus of the detection objective, because the detection objective also needs to be aligned with the illumination beam. So by default, Huygens assumes it's aligned, but actually here in the yellow channel, what we can see, if we move a bit here, we can see that all these yellow PSFs, they have a bit of a blur on the top, much more on the top than on the bottom. So it's a bit asymmetric. And that corresponds when the objective is focused below the light sheet. So I add 10 here. I see this dot moving down, which means which would cause this type of PSF. Now, I also here, I click apply. Yes. I can also make a PSF preview like it is here. Proceed. And I hope you can appreciate there's just a bit more intensity towards the top of the PSF than towards the bottom. So that's because we set this number 10 here. If I would increase this number, then it become even more asymmetric. So it comes with automated PSF generator for you to see that the PSF that Huygens is using is applicable to your data. So I go apply. Yes. If you notice, there's another question. If you notice the illumination is off center, can you adjust that at this time, please? Yes, you can do that. So if I understand your question right, your question is that if you notice that the illumination is not aligned with your light sheet, like is the case here, then you change that by changing this number, and then the red circle indicates the focus of your detection. I hope that answers your question. If I didn't completely understand your question, maybe you can reformulate it or let me know if that's it. In the XY, I see. So your question is if you notice that, so this is a little bit of a technical answer. So if you have a scanning system like a confocal system, then your detection and illumination PSF can be misaligned, and then you will have the effect that you're discussing about. If you use a light sheet, and especially if it's really a sheet and not just a single beam, but really a sheet, then you will have this effect because it's a sheet. Then if it's translocated in Y, it will be less, you will not see it unless, of course, it's a pencil. And then you can, there's the option to use the measured PSF in that case, if you don't have a lot of, if your light sheet is pretty flat. I hope that answers it. It's a bit technical for me to go in now in more detail, but if it's relevant for you, we're happy to discuss that later. Thank you. Welcome. Okay, so I set my settings. I'm going to close this, and so because this is still a work in progress version, it is not updating correctly yet, but it will. So I just need to make sure that I enter these settings that we just set. And this will, of course, be fixed in the final version. And I go accept. Now, of course, I can also save this metadata and then apply it in batch later. Okay, so these were the technical data that I need to enter as a user. For the other information, the Huygens deconvolution will take care of it. So I have here my image, and I'm just going to hit deconvolution express. And now using the metadata that I set, I will, Huygens will start processing this data. It is estimating the signal to noise ratio for each channel. It is estimating the background for each channel. It is divvying up the image along this direction in order to apply the spatially varying PSF. And it is also very good at whenever it is currently doing a calculation on a certain piece of image, it moves that image to the GPU, right? And if it's done with it, and it needs more space on the GPU, then it moves it back on the CPU. So Huygens is dynamically moving data from the CPU, so the regular RAM and the GPU RAM, in order to optimize the performance that is happening here right now. Yeah, so this makes it faster. So it's very good that even if your data doesn't fit entirely in the RAM of the GPU, it tries to make it into smaller bits that do fit on the GPU and then calculate it bit by bit with an accelerated speed. So that makes it very nimble as well. So we can see its progress here on the bottom. So it's currently, I think it's currently doing channel number 2, right? Because we have 2 channels. So the first channel is already ready. And the first number sort of indicates the overall progress, and the second number indicates the progress on the sub-task. So it's a useful tool to get an idea about how long a certain operation will take. And for this data set, we'd like to do the deconvolution, then the stitching with the vignetting correction. After each step that I now make on my crop, I can save a template that I can then use in the workflow processor to apply this to all the tiles that I have. And once I've established that it works well on this crop, I will then use the workflow processor. Before I go home, typically I turn it on, and when I come back the next day, I have my image deconvolved and stitched. And so in that way, I make Huygens work for me when I am actually sleeping. And that just works. That works wonderfully. So I think we're almost done. There we are. So I have a bunch of options now. I can save a report, and this will output all the information that Huygens used during the deconvolution. So for example, here you can see that the SNR is being reported and how many iterations were used, etc. All this technical information will be included in the report. I recommend you to save it. Then when you're writing your paper, you can have that information to include in your materials and method sections. Now we have different options here. The twin MIP for 3D side-by-side comparison, but I'm going to use the 2D, so the twin slicer tool. So here we can see if we look at the blue channel, let me just increase the contrast a little bit here. Is vignetting correction option included with the stitching module? Yes. Yes, so the stitching module includes the position optimization and the vignetting correction. And we're making a lot of improvements for the stitching correction to this release. So stay tuned for the next webinar. So if we look in the blue channel, then we can see here at the edges where the PSF is already getting bigger, we see a nice increase in the resolution for the blue channel. And what I really want to show you is the orange channel. And let me just crank up the contrast just a bit more. There we go. Here you can really see a big change. So this channel is then very much affected by the autofocus blur. And this is now a XY view. But if I go to the XZ, so I'm looking now again from the side, I can see here that what I was talking about, I have a very asymmetric PSF. And maybe I should even have used a more asymmetric PSF for deconvolution. And then in the result, that's just counteracted very, very well. And if I then just now hit play, this is an XZ view. So I'm now looking through the Y-axis, and I can see all these filamentous structures moving and changing, and with a good resolution. And you can get this for the whole image. Okay. So next, I want to show what's new with our pyramid file and big data. So with Light Sheet, the issue is that you have big data, which requires big computers to handle that. And so some don't have access to such a big computer, and that means that they're limited in the size of the light sheet that they can acquire. And others, they do have access to a big, powerful computer, but that computer often needs to be shared. It's located in one location, and it is not, you don't have access to it whenever you want. So we have now first implemented a pyramid file structure as part of our stitching module. And that enables you to stitch and deconvolve images that do not fit in your RAM. We still recommend that one tile can be deconvolved and that fits in your RAM. Otherwise, just a deconvolution, it needs a little bit of juice, but your whole image no longer needs to fit in RAM. So it reduces the amount of RAM you need to deconvolve an image. Thereby, you can handle bigger data with the images that you have. It also means that, let me just prepare that. Yeah, there we go. So this is now a different session of Huygens that I'm running on my desktop. And this desktop is a regular desktop. It has 32 gigabytes of RAM and 8 gigabytes of VRAM. So this is a nothing special desktop computer. And this is a pyramid file of 380 gigabytes that I can drag and drop into Huygens. And now I get to see a representation of this fly brain. So this is the fly brain of Dominique Villion that I was also showing earlier. So this is now, you can see the pyramid shown here. So this shows that the tip of the pyramid, so this is the lowest resolution currently being used. And if I wanna here open a 3D renderer, you can see that I'm now rendering that at an intermediate resolution. I'll just crank up the contrast. So it was injected with a fluorescent dye in its eye, and you can see the optical nerve moving through the brain here in 3D. So for the 2D viewer, so that I'm going to open the twin slicer, there I can dynamically update the resolution level using lazy loading. So this is the brain, and this is the bottom of the brain. And now I go up, and so these two bright structures, they are the optical nerves. And I go up, 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 up. And then here's a very bright structure at the top of the brain that I now want to look at in more detail. So as I zoom in, Huygens will automatically load the resolution from the appropriate at the appropriate resolution level. So I just now I was at an intermediate resolution, and I'm going to zoom in further to look at the highest resolution that we have. Here we go, and I can also look here. For example, look in other places, and we see a different morphology between these two regions, and that works. So that means that if you have a very big data set, you can combine it with Huygens everywhere. You want to look at it on your laptop, on your desktop, maybe it works on Mac, so you can use this in combination with Huygens everywhere to flexibly look at your large data and no longer be bound to a powerful workstation or powerful server. With that, I'd like to close. I will stop the recording. Thank you so much for listening.